I'd like to have distinguished Professor Fisher introduce our speaker today.
Thanks, Josie.
Yep.
Hello. Good afternoon. Yes. My pleasure to introduce Dan Madigan, today's speaker. Dan has been here as a research scientist, postdoctoral, NSF postdoctoral fellow position for a little less than 2 years now. He's got his bachelor's degree at Dartmouth in biology and his doctoral degree at Stanford University. He came here to do some work, initially continuing our work on Fukushima radioactivity in marine tunas. But as long as we had all these tuna samples in our freezers, we thought it might be an interesting thing to look at other contaminants in the tuna, most particularly mercury. And this is which might be the biggest, baddest metal in the biggest, baddest fish. And it's an interesting story, I think, that he has to tell you. It's quite fanciful. I don't know if any of it is real, but and he doesn't either. But he thought he would just make conversation today with you guys. So here he is.
How do I do the lights here? Is this working? No? All right. All right. Okay. Thanks, Nick. You said you'd insult me more, so that's disappointing. But and thanks, Josie, for letting me speak and for letting the word baddest make it through the screening process. I didn't think that'd make it, but it did. So on that note, I want to admit to my false advertising, the bait and switch. I hope some people came in here because that means it worked. To see something, great white sharks, megalodon, something about, the really biggest, baddest fish in the ocean. Like this one about to eat 2 whales. If that's your thing, it's okay, because Damien tagged Megalodon, Damien Chapman. I don't think he's here. I wanted him to... So that's what that is, right? Megalodon?
Yeah, that's a Megalodon.
Okay, yeah. So he tags those. So that's
the biggest, baddest. But
yeah. So you can talk to him if you're really into that kind of stuff. All right. So, but what did I mean by some of the fish that I'm talking about are some of the biggest? And what did I mean by baddest? I really, I guess, meant baddest in terms of mercury. So this is some data from a paper in 2007, and I apologize for the resolution. It's actually like that in the paper, so it's her fault, not mine. This is the highest mercury concentration in a variety of fish coming from imports, Atlantic, Pacific. And since you probably can't read it anyways, I just thought I'd put some pictures up there for you to see what some of these animals are. So up there, that kind of blobby-looking guy is tilefish. There's actually a fishery for them off Long Island. They happen to be really high in mercury. And then the other 3 may be a little more familiar: swordfish, shark, marlin. These are pretty well known as being high mercury fish. Okay, so you might have heard of these as being some of the high mercury fish, but there's another piece of that, and that's consumption, right? I mean, if some species is really high in mercury, but it's not consumed that much, it's not that, you can argue that it's not that important overall in terms of mercury going into your diet, into the diet of the entire population. So of course you can measure consumption also. So in this case it was measured in the US seafood market, and you get 4 new creatures there. Tuna, that's all species. That pictured as a yellowfin there, but that's all tuna species. Highest consumed fish in the United States. And then shrimp, pollock, which is your McDonald's McFish sandwich, and fish sticks, fake crab in California rolls, all that kind of stuff. And then salmon. So probably none of those are all that surprising to you. So the product of concentration times consumption is really what's going to give you the overall big players in the total mercury intake for the population. So we get a reshuffling now of the animals you just saw when you talk about total mercury intake, at least here for the US. And what you see is that it's tuna, swordfish, pollock, and shrimp. But tuna really kind of overshadow everything else. So I really want to point that out. And when you take tuna and swordfish together, you're already accounting for more than 50% of all the mercury coming into the U.S. diet. And tunas, swordfish, some other species, these are pelagic fish, which means they live in the open ocean. They're actually referred to as tuna and tuna-like species. That's a real designation. And what that designation means is tunas, billfish, fish with a bill, like swordfish and marlin, and also some other open ocean fish that you're probably familiar with, like mahi-mahi. So why mercury? Why? There's a lot of studies on mercury in all forms. So why? Why is it? Why do we study it? If you shop at a supermarket, if you buy fish, even if you don't buy fish, you've probably come across something like this, seen it in stores. And this is a warning for pregnant women, women of childbearing age, limiting mercury. And it seems to always focus on the same 4 species, which I find kind of fascinating: swordfish, shark, king mackerel, tilefish. I don't know about you, but I don't see, I'm sure parts of the country eat king mackerel, see a lot of king mackerel. King mackerel, shark, and tilefish are not species I come across very often on menus. They're not species I come across very often in the seafood department. And I'm always surprised that tuna, at least the big tuna that we know are high in mercury, are not included on this list. And I think there are political aspects to that too, but I don't know the details. So again, why mercury? So we know that mercury can affect human health and that it affects the nervous system. There's a lot of debate about how much you need and how it affects certain individuals differently than others. But we know that at a certain amount, it definitely has deleterious effects. And again, there's a lot of debate as when you get below some threshold, does it matter at all? Effects become hard to measure. I'm not going to get into those debates, but it's clear that from all that research that there is a concern out there about consuming mercury. And again, especially for pregnant women, because it can affect developing fetuses. Just a real quick look at where this mercury is coming from. Right now, the main sources are coal burning on a global level. And the reason coal burning affects on a global level is because it puts mercury into the atmosphere, and then the mercury is subject to atmospheric circulation before it gets deposited into the oceans. So when you have a lot of coal burning in one place, that doesn't mean mercury pollution in that one place. It means it can spread all over. And there are more localized activities that lead to more localized effects. One of those is artisanal gold mining. A lot of silver is released by this process, and local runoff will make potentially high mercury concentrations in those local aquatic systems. And then finally, just a quick look at the types of mercury that are out there. When it's first released, it's in its inorganic form. It's a metal. It's mercury. Through biological processing, a methyl group is added to that mercury, and it becomes methylmercury. Why does this matter? Well, methylmercury is the one we're thinking about and talking about when we talk about human health effects. And what I think is really kind of some amazing numbers is that in aquatic systems, methylmercury really only accounts for about 1 to 2%. But once you get into fish in those same systems, it tends to account for around 95 or more % of the mercury in that fish. So why would that be? The inorganic form is both absorbed less readily and excreted less readily. So a product of those two things means that inorganic mercury can come in, but it can also leave fairly rapidly, whereas methylmercury, once it's in, it wants to stay. It's a simplified look at it, but it's generally how it works. So for me personally, why mercury? So I study pelagic fish. I'm interested in their ecology, in their movements. And this is a paper that came out very recently. And they're kind of trying to look at why certain pelagic fish, again, open ocean fish, have higher mercury than others. And they used what these colors refer to as depth. So you have depth on the y-axis, surface is the top, and on the y-axis, I think I said, and on the x-axis are certain isotopes of mercury. And those isotopes reflect the depth at which the mercury is acquired. So what they basically found is that as you go into deeper diving predators, the predators that spend more time at depth, their mercury signal is more of, it reflects that they're acquiring the mercury at depth. Okay, that makes sense. And then kind of a companion paper with some overlapping authors showed that through things like archival tags, which can track what these fish do in their wild environment, that the median depth of occurrence, basically the deeper overall the fish spends its time, the more mercury it tends to have. So what this study suggested is that surface feeding predators like yellowfin tuna and mahi have lower mercury, swordfish have higher mercury, and this has to do with the depth of feeding. And this tied in pretty well with this. So the combined, this kind of says, okay, deeper divers access higher mercury prey and thus have higher mercury. But based on some of the stuff I had done, I didn't really fully buy this story. It didn't seem to fully make sense, and I'll get into why that is later. So this is some of the work I had done previously using stable isotopes to reconstruct the diet of pelagic predators. And if you can't really see understand or recognize these icons, these are some of the major players we talked about earlier in terms of pelagic fish that have high mercury. Some tuna species, sharks, swordfish. And we had all the samples from this study. This was, a few thousand samples from the predators and also their prey. Now, if we've reconstructed the diet of these predators and have a general idea of the inputs of the different prey into the predator diets, and we can also measure the mercury in that prey and the mercury in the diets, we can start to get an idea of how much the diet mercury is dictating the mercury levels we see in the predators. So that was kind of the idea, to just get going and start generating data. And at the same time, we have an ongoing collaboration with researchers in Taiwan and Japan. These are pictures from Taiwan. This one in the upper left is me sampling with our collaborator Riyar at a pretty typical fish market there. This is where the fish come when they come off the ships. And you can see there's some mahi here. We're measuring a blue shark. There's a big marlin here. And all those buckets back there are just filled with mahi. So this is a typical single day. Those fish will be gone in an hour. And you could come back to that same spot in 2 days and do the same thing all over again. So what we really did is acquire thousands of samples of all these different species from one place. And what's kind of sad about the fishery, but makes it useful for this type of study, is that they don't really have size limits. They have access to the big fish. The big fish happen to go through there. But if they catch a swordfish this big, they keep that swordfish. Probably not great from a sustainability standpoint, but and it'll be interesting to see, honestly, how this fishery plays out because it's so active. But from a research standpoint, to be able to access that range of sizes is pretty unique. I just want to point out, this is a giant bluefin coming in. They all get very excited when a big bluefin comes in. It's like everyone gathers around. You know something big's about to happen. They don't care about like a 800-pound marlin coming off the boat. That's not a big deal. But if it's a bluefin, it's a big deal because they're so valuable. And this is one way that they catch marlin there. They actually sight. This is a spit off the front of a small vessel. They'll sight marlin in rough weather like that and stick it with this 3-pronged harpoon, and that each prong is attached to stainless steel cable. So they tug that thing in over time. So all the fishermen are missing fingers and stuff from doing this kind of thing. Okay, so I'm going to take you through kind of how this study worked for me and for us. We took preliminary measurements just to kind of see what was going on and get the data in the first place. And then we started, we had access to captive tuna, which I'll talk about more, which really helped kind of set the framework for going forward with looking at the wild data. From the captive tuna model, we wanted to modify and change it to make it useful to other scenarios, and then apply the model. And the reason I have the question mark there is we weren't sure if this would work, how you can apply it to wild fish, but we wanted to try. So first I'm going to show you preliminary measurements. That's at one shelf in my office, the daunting tower of samples that needed to be processed and measured individually for mercury. So I just want to show you a few of the kind of, as data was coming in, what we were seeing that seemed strange based on very basic expectations. So there's kind of a paradigm out there, big fish, high trophic level, high mercury. If anyone is not familiar with the term high trophic level, it just means high on the food web. And generally, the bigger you get in the ocean, the higher you are in the food web. So here's a pretty big fish on the left. That's a mahi-mahi or dolphin fish. And there's a mackerel on the right. And we took measurements of some of these, and the mahi were pretty surprisingly low in mercury. And we found that consistently these mackerel were higher than the mahi. And this seemed strange, especially since I know and have seen mahi eating these guys. So right away, how does that food chain part tie in there? What's going on? That seems strange. So like I said, we had a low mercury predator eating a medium mercury prey. Seems weird. And then we started measuring swordfish, and swordfish were like, you know, the crazy numbers. The kind of, if you can call it exciting when you look at the machine as it's working through each sample. You know, it had to clean itself after some of these swordfish samples. They really were very high in mercury, and there's a decent amount of diet studies for various regions. And swordfish eat a lot of squid, and when we measured squid were some of the lowest mercury prey we were finding. So again, we had one of the lowest predators eating a higher mercury prey, and one of the highest predators eating a low mercury prey. So again, this was confusing. We wanted to look at size as well as part of the big fish high mercury paradigm. So we had a wide range of sizes, as I mentioned. So 50 centimeter mahi, something like that, and 150 centimeter mahi. That's a pretty big dolphin fish. That's like a dolphin fish that's about to die of old age. And it increased about 30 times. Is that high? I don't know. It seems high. It's all relative, right? So it seemed like a big increase. So okay, that makes sense. Same species, bigger fish, higher mercury. What about a different species? 70 centimeter swordfish. In terms of what should be caught and what is usually caught, that's a very small swordfish. But it's still not a small fish, you know, 70 centimeters. And that went up. Our biggest swordfish was about 290 centimeters. It's getting pretty big, almost a 3-meter animal. And the mercury increase there was 450 times from a small to a large swordfish. And I was pretty confident this is a lot. That's a big increase. And what I thought was kind of surprising, to put it in context with you know, humans in the picture, that's about those sizes being held by, or with people next to them. And while I would agree that this is a very big animal compared to that one, you know, these aren't like sharks. They don't take bites out of big stuff. They are gape limited. They swallow their prey whole. And when you look at studies that are out there, you know, diet studies, they really do eat the same things. There's no doubt that bigger swordfish can swallow bigger stuff. But really, they love squid, or in a certain area, they may love some other prey. And while it could eat that prey at larger sizes, the larger swordfish, their diets don't change that much. So a 450-fold increase I thought was pretty amazing. Okay, well, swordfish are a lot bigger than mahi. So what about a predator that goes through similar size changes? We measured yellowfin. We had a yellowfin at about the same minimum size as those small swordfish, and the yellowfin that get reasonably large, reasonably close to that large swordfish, they only increased 15 times as opposed to 450 times. So anyone who's familiar with mercury, or just manages to think of this on their own, in fish, are probably wondering, you know, when are you going to talk about this part, the growth rate part, that fish get older and they accumulate mercury over time. Okay, so swordfish have a slower growth rate, yellowfin have a faster growth rate. Does that explain it? Well, what's weird is that at the same size, so obviously if they have a different growth rate, it should affect it throughout their sizes, theoretically. The yellowfin had higher mercury at similar sizes, at small sizes. And then once you got up to the big ones, the swordfish had gone way beyond what we saw in the yellowfin. So this kind of brought us to the most important scientific question of all, in my opinion. What's going on? What are we going to do with this? So, as I said, you know, those were our preliminary measurements. This was the stuff that kind of gave us insight as to what we're seeing. Now what are we going to do? So we were lucky enough to have tissues from captive tuna. Having these pelagic fish in captivity is extremely rare, and it's really a valuable opportunity that I was able to work with these. So these were held in captivity at the Tuna Research Center at Hopkins Marine Station, which is part of Stanford. And these fish are fed the same thing over time for long periods of time. And as I said, it was really great just to be there. Anyone who fishes here or studies these pelagic animals, to see them behave. Obviously, a tank is not wild conditions, but to see how they behave and the differences in behavior and just watching them feed is really pretty cool. So if for nothing else, to wake people up if they're tired and bored, I put this in. I'm going to use the mouse. This gives you an idea of what a typical feeding is like there. The music's funny. I'm not allowed to change the music, okay? This is TRCC mandated music. So feel free to, pull out the light sticks or whatever. This is the calm before they're being fed. So now we see mahi and tunas getting into it. I think it's kind of fun to see. At certain points, you'll see the prey fall in. And if you watch it, their efficiency in plucking them off without hitting each other, it's really pretty amazing. There's a scared mola. He has to get fed separately. He can't keep up with the other guy. So what's cool about this is they're fed a very specific and consistent diet of squid and sardines, and they're in the same ratio all the time. Not only that, the mass of food relative to the mass of predator is always kept constant. So they keep track of the predator's mass in the tanks and make sure that at certain times they're not overfeeding and other times underfeeding. That's the finish of that song.
Excuse me, did these guys individually, did they ever get satiated, or to somehow compete the others and eat more, or?
So there's, in the beginning, it was obviously a learning process to keep them in captivity. And at first, they were feeding them to satiation often. And the tunas were actually dying, and they didn't know why. And when they did necropsies, they had so much fat in their hearts that they were having heart failure. So that was on an all-sardine diet. That's why they switched from sardine to sardine and squid, because squid are lower lipids. And this is something I was interested in doing, but it's just like such difficult work, and I just didn't manage to get around to it. Behavioral stuff like that, dominance in the tank. And these fish do have colored tags on them, and you can follow. It's hard. You see how fast they move. But you can go above in the rafters and say pink, 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 blue, blue, yellow zebra, yellow zebra. That's the yellow and black tag. And you see that there is a sequential dominance in these fish. Some wait around. The squid go first because they like those less, and some wait around for the sardines to come. They've learned that the sardines come, so they'll wait until the good prey comes. Pretty interesting. So again, this tunas in captivity situation is really pretty unique and pretty useful. When I got there, it had been going on for years, and every time a fish was sacrificed for experiments, tissues were kept in the freezer in a -80. And what we had were not really intentional, but what we had were samples from captive feeding experiments. That's really what part of this is that they're fed the same thing over all that time. It's a captive feeding experiment. And they range from 1 day in captivity to 2,914 days in captivity. So that's about 8 years. So that's a pretty long experiment if you were going to start it from day one. But it was already right there. And as I mentioned, what's really cool is that you can measure, they have consistent food, you can measure the mercury in the food, you can measure that Hg0, or first, initial Hg in the fish, what do they look like when they come in. That's going to change over time, and at any time t, dependent on Hgt is at mercury at time t. How long they've been in the tanks, you can see what mercury levels they have. So I'd use this for stable isotope turnover studies. What you basically have here is carbon, stable isotopes of carbon on the x-axis, stable isotopes of nitrogen on the y-axis. They came in looking like one thing, their food was different, and they ended up looking like something else. And over time, there's change. And you can get turnover times. How quickly will they turn over? What do the dynamics look like of change of these different isotopes? So that specifically for nitrogen-15, over time, this is what it looked like. And you may recognize this if you read my last talk, because I showed this also. This is 2 tissues and muscle and liver, as you can see. And we just made an exponential fit. You can see how long it took, 500 days, reasonably long time, depending on how you look at it, to get to some sort of steady state. That's basically what we saw, change, right, in response to food, and then steady state conditions. So it seemed like we could just do the same thing with mercury, just substitute mercury in. And without really knowing what to expect, I had expectations anyways, and that was that there'd be some change and then some steady state. If you've ever heard the term biomagnification factor, this idea that you're 4 times higher in mercury than your food, that suggests inherently that some balance is reached between you and your food, right? So I figured there'd be some asymptotic value. Okay, so here it is, same fish, mercury now on the y-axis and time in days on the x-axis, and this is what we saw. And I kind of was like, ugh, I don't know how I feel about that. I mean, it was interesting. We saw definitely a decrease, a lot of noise, a lot of variability, and this is what I really wasn't sure about, was this stuff. Yeah, you can fit an exponential model to it and get an asymptote, and I wasn't really sure we were seeing an asymptote. It's obviously a little unfortunate that the further along you get in time, the more rare those specimens are. You know, you don't get that many fish that spend that much time in the tanks. But I did notice the outliers in the middle were low, and the outliers towards the end were high. So I wasn't sure exactly if we were seeing a steady state with a lot of noise or something else. So finally, we actually read papers and looked at what drives mercury dynamics in fish. So growth's a big one, and very simply you can show it as growth dilution. So what this basically means, and this is kind of hard to describe very quickly because parts of it are counterintuitive, but basically mercury wants to stick around, okay? So in an animal, anabolic, breaking down of tissue, turnover of tissue, and catabolic processes are happening all the time. So as you're recycling tissue and taking in food, so let's use a person as an example. If you're maintaining a steady weight, you're taking in food, you're taking in more mercury. Why are you maintaining a steady weight even though you're taking in food? Because you're using other tissues inside, breaking it down, turning it over, using it for energy. The toxins that come in stick around. So as you're catabolizing tissue, the mercury doesn't leave with it. So the more you can tip the scales, catabolism is always going on. The more you can tip the scales towards anabolism, the more fresh tissue is being made, and the more fresh tissue is being made, the more overall you're going to somewhat reduce your mercury concentration. That's the best way I can describe it. So the easiest way to describe this is growth dilution, simple equation. An initial Hg concentration times e to the negative g, that's growth, growth rate constant, and time. And you're going to get a decrease. This is just an example. It doesn't have to be this steep. It can be barely noticeable, but you'll get some decrease and then some leveling off. That's what's important. There's another piece. It's just linear accumulation over time. You're eating, you're taking in food and taking in mercury. And as this is happening, based on what's been seen in other studies, for whatever reason, it tends to be a linear increase. So what are the terms of this? You have some uptake rate constant. That's a net, right? So that's not just uptake. It's taking it to everything. You uptake at a certain rate. You excrete at a certain rate. The net will be your uptake rate constant. Food's obviously going to matter. You're going to multiply that times the amount of mercury that's in your food. And then if it's a function of time, if it's a function of time, time's going to matter also. So what does it look like when you add those two curves together? You get something like this, right? This matters a lot early. And then as it levels off, the time-based accumulation is going to take over. And when we applied that, when we went back to our captive data and applied that, it fit better. We were happier with what we saw. The variability didn't go away because the data are the data. But we really felt confident that this is explaining some of what's going on. You can see the R-squared was about 0.5, explaining about half of what's going on. So I want to just point out that, okay, this is the raw data. It's for 64 individuals. And that's how we fit the model. And we had to assume the same growth rate for all fish. Remember, growth rate is part of the equation here, and that was just a number. Since these fish were in captivity, this is something you couldn't do in the wild. Since fish were in captivity, we could actually measure individual growth rate. So one question was, how much does variability between individuals and growth rate affect this? How much is that accounting for the noise? So we could go back in and now use that equation to make estimates on the mercury levels that we'd expect based on just time in captivity and individual growth rate. When we did that, things got a lot tighter, and it seems like that variability in growth rate adds about 25% more of explanatory material for the data. Another thing I thought that was interesting about this is some of the weird points, some of the real outliers, you kind of saw in this also. So this, for example, would probably be two fish that were growing a lot faster than the other ones in captivity. You remember that, with that decrease, these decreased more because they were growing faster than the others. Maybe they were eating more, maybe they were healthier. So this was using the model to estimate the mercury based on actual individual growth rates. And before I move on, I just want to make sure that people are solid on what we're looking at here, because the decrease I've found is confusing to people. You don't expect a tuna this big, when it becomes this big, to decrease in its mercury. So again, what we're seeing here is we brought fish in from the wild, started feeding them prey that they would maybe eat in the Eastern Pacific, sardine and squid, and then they decreased, as you see, and then they started to increase again. Now, that could be the way we saw it is it could be due to two things, two major things: faster growth in captivity or different food. And the TRCC actually makes a point to mimic wild growth rates so that they avoid that being a confounding factor in the kind of the stuff that when they're doing experiments. So if growth was the same as in the wild, and we don't expect them to suddenly decrease in the wild, and based on other studies of wild fish, you don't tend to see these sudden drops. We assume that the food in the TRCC was probably lower than what they were eating in the wild. And maybe if they had come in and kept eating what they had been eating, we'd see something more like that. So from fitting it to the captive data, we obtained something important. We got one parameter, this uptake rate constant. We couldn't have guessed that or had that if we didn't have these tunas in captivity. Now, how do we modify this model for natural conditions? How do we modify it for what they might really be eating? And more importantly, in captivity, they're eating the same thing constantly. We know that in the wild, these fish may go through a trophic increase, a trophic change, eating different things as they get bigger. How can we incorporate that if we're going to look at wild data? So now we're up to modifying the model. Does anyone know what that picture is? That's an airbrush. Modifying the model. Anyone get it? Just think about it. It's a risky joke, I know. The model, where we're at, the model, if we know time, which we get from age, which we get from size, we know the growth rate, we know the food inputs, we can then estimate the mercury dynamics over time. Okay, so what are some of the challenges to changing it? Different pelagic fish have different growth rates. What do we do about that? Well, there's lots of age growth studies on pelagic fish, and luckily the von Bertalan land feed growth equation has become basically a standard. So even as they've improved on it, that's almost always in every age growth paper. And they have a growth parameter, K. And for all these pelagic species in specific regions, we can take that K, we can take the K for Pacific bluefin, and we can just scale the K. So if a mahi-mahi's K is 3 times that of bluefin, we just change the growth rate parameter by 3-fold. So we can modify it for a specific species. Okay, different fish eat different things, so that's pretty easy. We just change the food input. And then we know that food can change over time. This is a little trickier. And what I mean by this is, as I mentioned before, as you become a bigger fish, you just have more options. And it doesn't mean that you just eat bigger stuff necessarily, but it means you can. And in the wild, that has to be taken into account. So what we did is we put in this trophic level factor that's multiplied times the food. So now instead of having a stable food mercury concentration that's throughout the life span of this animal as it gets bigger, it allows for it to gradually increase. And I just want to point out now that once we applied the model, this factor could be 0. We gave it complete flexibility. So we weren't assuming that this definitely happened. We just gave it a chance to happen mathematically. If having some trophic level increase made a best fit to the data that we saw, then that's what happened. If a trophic level factor of 0, in other words, their diet isn't changing much through time, that's what we got, was 0. So we didn't have to assume a value for this. We just let it be open to the model estimate. So this just let us start to play with different variables and see what would happen in different scenarios. Total theoretical. So in case you're wondering, that captive bluefin model, that's all the way down here. Why isn't it like this anymore? We had to change the y-axis. So the y-axis scale now is way increased, okay? So that's what the bluefin in captivity were doing. What happens if we have the growth of that bluefin? Not much. They didn't get that initial dip, right? But it's pretty much, for all intents and purposes, the same. Once you started to double food, you saw changes happening, not too much in the beginning, but over time, due to that linear increase, it changed. You can add just a trophic level shift. Thought that was pretty interesting. it's pretty similar to doubling food, and at some point it exceeds that. You start to get a more precipitous rise, double food and add a trophic level increase, and then kind of do everything you can to increase mercury. Half the growth, 4 times growth rate, quadruple the food, which is not unreasonable actually based on the numbers we saw, and add a trophic level, and you see just this precipitous rise. But again, this is just messing around with variables to predict how these, or to look at how these different variables interact. So what about real data? So now we want to really, I started to see this now as a tool to understand things about wild fish, and I thought that was pretty cool because, you know, if you do ecology research, a lot of times you end up putting out a paper and it has interesting information in it. I'm not used to building something that I can actually use for anything, you know? Like engineers get to do that. Felt nice. So now we have a tool that we can apply to different things and get something. But this was a question too. Will this work to apply it to all these other species? As I mentioned, we now know how these variables interact, right? That's really important, that growth makes a difference. Food makes a difference. A trophic level shift makes a difference. But they all interact in a certain way. So you can't say one makes a bigger difference than another. It's an interactive process. We have that in the equation. What can we put in for these different species? We can change the growth rate, as I mentioned, modify it for the species. We can get an initial mercury value. That's basically take your smallest fish for each species, and that's your starting point. You know, we don't start with larvae. We started with what was available to us. Generally, for almost all species, something in the range of this. And that's Hg0. We measure that. And then you can get the time since Hg0 simply from the size of the fish, back calculate its age, and see how many days it's been since it was the size of the fish at Hg0. What don't we know for these different species? Well, we don't know how their diet differs from the others, and we don't know how their trophic changes over time. So these are really ecological variables that will help us explain what's happening in these different species if the model works. So I just want to show you first now it's important to note this is size, okay, not time yet. This is size on the x-axis and mercury on the y-axis. So I just want to give you an idea of what some of these fish look like. I thought it was pretty, in certain ways, pretty amazing. Here's this dashed line. This is the FDA action limit. I don't even know what that means. I don't think they really know what that means. But they decide, it makes no sense because they decided that a certain value shouldn't be in the markets. A certain value of mercury in fish shouldn't be in the markets. If you eat a very small portion of that once or twice a year, it matters a lot less than if you're half the action limit and you're eating it every day. So, you know, this is probably not surprising that they have a single number that matters. But anyway, that's the number. And you can see that for all the species, up to a certain size, they don't cross that threshold. And then they all cross that threshold at different sizes, besides yellowfin, which never crossed the threshold. And if, you know, obviously this guy kind of stands out, and that's swordfish. But in terms of applying the model, the logical place to start, we built it on captive bluefin. Let's start with wild Pacific bluefin tuna. So here we've now taken not size, but time. We took that size, we converted it to age, and we did time versus mercury in Pacific bluefin. R squared was 0.6. You can see there's still a lot of variability, right? I was pretty happy with that fit. No line is going to fit data like this that well, and the variability is interesting in and of itself. But one thing that's interesting is, I just want to point out, because we'll talk about this briefly later, is a lot of the outliers were in that middle time frame, and those were abnormally high. Okay, but what do we get out of it? Prey mercury is about 0.24. That's the prey mercury concentrations that best explain this pattern. And when we measured prey in the Western Pacific, which we had, they fell within that range. So at least, you know, things were making sense. This didn't make make zero sense to us. It also, the model said the trophic increase was negligible. We went into some studies. It turns out that bluefin kind of stick with what works. A bluefin this big and a giant, with some exceptions, time to time, will feed on energy-rich schooling fishes. Sardines, anchovies, fish like this are abundant enough in both the Western and the Eastern Pacific Ocean that a giant bluefin can get by. They'll eat more of them, but they can get by on these small, nutrient-rich schooling fishes. So then we applied it to everything. Okay, it's kind of a lot. One thing important to point out here is that It's time on the x-axis and mercury on the y-axis, but the scales aren't all the same. It's fish specific. Here, the dolphin fish, the scale of the y-axis is up to 1. In swordfish, it's up to 25. So there's a big difference there in the maximum values you're seeing in the different species. So now, one of the goals of this, because a lot of people compare, they assume that swordfish is higher in mercury, it must eat higher mercury prey. Dolphin fish are low in mercury, they must eat low mercury prey. The model, again, gives that as an output. So what I was really interested in what it told us about what prey mercury values explain what we saw. And I want to go back to this real quick and just bait you a little bit. You look at this graph, there's the swordfish, and I'm going to use as another example, the blue marlin. Which of these species eats the highest mercury prey? Come on. You look at that graph, what do you say? Simplistic answer. Yeah, swordfish, okay, thanks. And especially if you're going to compare it to blue marlin. Blue marlin's somewhere in the middle, okay? So you'd assume they're just not feeding on a level that's as high mercury. And really what our model showed was that Pacific bluefin, striped marlin, swordfish, and yellowfin, very similar prey, explains the numbers, mathematically explains the numbers that we saw. It doesn't mean they're eating the same thing, it just means that they're feeding on prey with similar mercury values. And again, that's especially surprising, I think, when you look at this graph. That's not intuitive. And the two species I want to point out, because they're different, are the blue marlin, which fed on prey, or it was estimated to feed on prey about 4 times higher. And again, there he is over there. So that was interesting. And the mahi feeding on the lowest food. That made more intuitive sense. So again, we want to just dive into the literature and see if this just at least makes moderate sense. Mahi like flying fish. They like, this is called mene maculata. They like these things. And the range was right. Was this quantitative? No. It was just the range of fish. We took the range of fish mahi are known to eat. We got a range of values. It fits within that range. The blue marlin was kind of more interesting because it was relatively so high. And also it had a high TLF, that's trophic level factor, meaning it seemed to increase a lot in its trophic level over time, change its diet over time to bigger, high mercury things. And when we went into the literature, the one study on blue marlin feeding off Taiwan and Japan has it eating basically entirely skipjack tuna. And we had skipjack tuna, and we measured skipjack tuna. We found an amazingly high kind of concordance there with the estimate of the model that skipjack tuna were right on that number. And as far as the high TLF in a companion study, we're doing stable isotope work on these animals, and this is data I've generated with stable isotopes of carbon and nitrogen, and basically consider the red to be the big pelagic predators, and the green to be smaller, kind of inshore stuff. And you see as the marlin gets bigger, they're eating more stuff. That's what the stable isotopes said. So basically this totally separate analysis using stable isotopes was in complete agreement with the mercury model that blue marlin, as they get bigger, they really do take advantage of their size and increase the size and thus the mercury of what they eat. I just wanted to touch on the variability real quick because that's kind of a cool side piece. Four out of the 6 of these species showed the same kind of thing, this cluster of high values right in the middle. And it turns out pelagic fish, males and females, have different growth rates. And what we really saw is that once they started getting to their maximum size is really when you saw a precipitous rise in mercury. So you don't even see, because this fishery that we've been in, probably a product of the fishery itself, luck, and what's happened to pelagic fisheries, we don't get the biggest fish in the ocean. They say blue marlin, black marlin can get to 2,000 pounds. The biggest one we probably had, 800 pounds. I mean, that's really big, but it's not getting towards the end of the life of a female. Those are the females, of a female marlin. But the males max out earlier. And when they max out is when their relative growth decreases the most. And when their relative growth decreases the most, their mercury increases the most. So what we found when we compared where this was happening in terms of time, in terms of the age of the animal, This corresponds to males kind of maxing out in size. So what you're really probably seeing here, and it would be great to have male and female, to be able to know which of these fish are male and female, is two curves, a combination of two curves, a male curve and a female curve. If you could extrapolate that black one out, it probably goes up again at the end, but we just don't have data with those giant fish. Males are maxing out there, starting to get high in mercury. And when we looked at the sizes for males, that fit for all those species. So that's probably what you're seeing there, and that's why that noise falls in the middle. Okay, so we had all this. It was pretty cool. And then I was thinking, this is all the Pacific. Would this work in a different ocean? So logical place to start was another bluefin species. This is Atlantic bluefin tuna. This is from a paper in 2005. Tagging data, you can see they go all over the Atlantic Ocean. We were lucky enough to have these samples. Chen Li, who is a graduate student in Nick's lab, analyzed a lot. I should have put the N here. It's like 1,200 samples here. And I fit the model to this data. And first of all, it fit fairly well. The confidence intervals were tighter than we saw for a lot of the fish in the Pacific. And interestingly, it gave us an estimate of food that was very different from the ones in the Pacific. It was just spitting out the same number all the time. I don't know how I'd feel about it. But okay, I got a different number and no TLF. It was saying that bluefin do the same thing throughout their lives in terms of the prey they're eating. And again, dove into the literature, tried to see if this at least, qualitatively made sense. And when I found the bluefin again in the Atlantic, they stick with what works. They like oily, abundant fishes, mackerel, sand lances, menhaden. And when I looked at the mercury levels of those, their average was 0.11. So again, pretty amazing concordance here. And then there was another study that looked at bluefin from 50 centimeters to, I think, 1,000 centimeters. 1,000 centimeters? 100 centimeters. And they're basically going from footballs, as they're called, to giants. And the prey size didn't increase in bigger fish. Once in a while, a big bluefin would eat like, a spiny dogfish or something. But for the most part, they're eating the same stuff. And again, those papers supported what our model suggested for Atlantic bluefin. And I thought that was pretty powerful that it worked in an entirely different ocean basin. Okay, I want to quick go back to just this mercury at depth question. I questioned it before. This idea that the deeper predators are accessing this higher mercury prey environment. Does our model support that? And it's no. That a similar range of prey mercury levels is feeding all these predators. It's a product of something else. Now, if swordfish and yellowfin are eating similar mercury prey, based on our model, what would be the factor that's most likely to be driving this largely? Growth. So, in my opinion, this was a product of slow growth. So I took all these species that they measured in this paper, and for the ones that it's available, I looked up their growth rates. And it turns out there's a correlation between depth and growth. So what I think is going on here is when you're simply making correlations, you can measure mercury in a fish and then measure its depth of occurrence and just see if they correlate, and that's great. But I think there's a correlation between, based on what we found, there's definitely a correlation between growth rate and mercury. And if there's a correlation between mercury and growth rate and depth of occurrence, then you're going to get a correlation between mercury and depth of occurrence. And it's a spurious correlation. Okay, so I don't know if any of you run into this question often, but I do. So what? Okay, we found some cool stuff. It explains more what happens in these big fish, but is there anything more we can do with it? Is there any further understanding we got? The mercury's still in the fish, and people are still eating the fish. One thing I think that's kind of, depending on how you look at it, it's kind of cool, it's kind of weird, it's kind of bad. Every one of these data points, all these values you see, have now been consumed. We collected all these samples at a market, right? So, and it was a while ago. So all these high levels, including here's your action limit, here's this one swordfish. That swordfish has long been eaten by a human somewhere, and the mercury in that swordfish is sitting in probably lots of different people. But every single one of these data points have been eaten. So one thing we saw that we thought was pretty cool is the curse of longevity. Getting old sucks, and it definitely is bad for these fish in terms of mercury. The typical harvest range for the fast growers also tend to not live as long. So what we saw, based on the theoretical projections and also just how the projections look if you extrapolate them, is that these small fish, the fish that you consider, the pelagic fish that are considered lower in mercury, mahi-mahi, skipjack tuna, yellowfin tuna to some extent, if they lived longer, they'd look just like the other species eventually. They'd get to that point. They just don't live long enough to get there. So this harvest range of fishes basically catches them in this sweet spot before they really have a chance to get up there, based on their life history. On the other hand, for the slow growers, you have a huge harvest range. They're harvested at all different sizes. So a bluefin may be harvested down here, it's all right. Bluefin that's harvested up here, very high. Same with a swordfish. And one idea we had, which is definitely, unlikely, is you could take this in conjunction with good fishing practices and kind of maximize some way you could both minimize the mercury, pick size ranges where you're minimizing impacts on the population of the fish and minimizing the mercury that goes into the population. In other words, probably leave the small ones alone because of conservation concerns, probably leave the huge ones alone because you can let them breed, and stick somewhere in the middle where there's a little bit of a sweet spot from a conservation and mercury standpoint. So I still say, so what? What else could we do with this? One idea is linking human activities to mercury in their seafood. So this is a map of deposition of mercury in different parts of the world. And one really interesting idea, and people have tried to do something like this, is how do these different deposition rates in different areas correlate to the mercury they get in their seafood? So for a pelagic fish, what would you do to do this? You pick a couple regions, and you pick a species that exists in all of them. So yellowfin, someone I know calls them the rats of the open sea. Like, they seem to be everywhere. Yellowfin, you could get those from all these places I point out, okay? But there's a problem, and people have done this. They've done these correlations. But one issue is size, right? These studies talk about how yellowfin from area A versus area B are higher, and that's because it's higher in mercury. Well, you can think back on this graph and see how important it is, the size you pick, how important it is for you to pick the right size. As I said, swordfish up here, so the swordfish that we measured, the smaller swordfish we measured were some of the lowest values we found, period. Some of the swordfish values, a swordfish this big had a lower mercury value than a flying fish this big. So the size you pick matters hugely, right? So, okay, some people say they correct for size. You can see it's pretty messy, but you can fit curves to it, and the R-squareds aren't bad. So I see how that could be done. I don't think it's the best method in the world. Beyond that, you could actually collect the exact same sizes from all the different regions. You can imagine why that would be pretty tough. Even if you do, there's another problem. A yellowfin here doesn't have the same growth rate as a yellowfin here. They have different growth rates in different parts of the ocean. So how do you account for that? You really can't. Before the model that takes all the pieces into account, there's no way you can just correct for all these different interacting variables. And the last thing I'll mention is the manipulation of captive conditions. I have mixed feelings about this whole bluefin farming thing, but it's definitely becoming really popular. And it's happening in different countries all over the world. And basically, they can manipulate other parts of this. They can manipulate the food for sure. They can manipulate the growth rate by how much they feed. And you can imagine that it's pretty compelling to be able to put a tuna on the market that you can advertise as having 1/4 the mercury of wild tuna. So in other words, you can control the mercury and make a choice onto what you choose to feed them, and then control the mercury out. And what that would really equal for these places, obviously, is profits, which drives everything. So finally, the last thing I'll say is, I think this equation, I keep calling it a model, it's really just an equation. It allows you to assess old data and data coming forward. You can go back, and if there's size information and mercury information on any species, you can really more get at what's driving those differences. you can see if they're really, if prey in the Mediterranean, which is thought to be a more enclosed, polluted system, versus the open Pacific, just because one species is higher in mercury than another, that doesn't really tell you definitively that much. You can actually see if it's coming from what they're consuming. And then from there, you kind of have to go below that and see if why the prey is higher in one region than another, right? And that's a whole other piece. But it really allows you to look at this stuff comparatively in a much different way than just making simple correlations. And with that, I just want to thank you guys for being here, all these people who are both part of the project or have just helped generate data, and also the funders that have helped us do this work. Thank you. So I guess questions?
So the Fisher Lab has a long history of modeling mercury accumulation and looking at growth and growth pollution. And one of, probably the most common model that's come out of that lab is the biokinetic model.
Mm-hmm.
And you're using a different model, and I'm just curious if you looked at the biokinetic model, or if you chose this one in particular, if you've done any comparison in terms of whether they give you different answers.
Yeah, no, I haven't. This was based on what I found in fish, and I didn't want to use Nick's stuff anyway, you know. But no, this is based on what I found in fish. I haven't tried the biokinetic model, to be honest. I don't. What are the additional variables that adds?
One of the things that it looks like, if I were to kind of call up that model that you used in my head, I think one difference is that it includes things like consumption rate,
Okay.
Which is obviously really difficult to measure in fish, but it still allows you to make some predictions on what the mercury content is based on things like growth rate, and also growth efficiency, which is kind of, when you boil it down, that's the big driver. And that's kind of a second question to have, is if you have these fish in captivity, maybe you have data that you can calculate growth efficiency. In other words, the biomass in the predator relative to what they consume from their prey.
Mm-hmm.
Because what you're saying about growth dilution is really about buildup of tissue versus buildup of mercury and how those relate to each other. That's the growth efficiency is really what you're talking about in terms of growth dilution.
Yeah. And that'd probably be valuable because.
So you might get tighter relations.
Right.
If you looked at that.
Yeah. I mean, one of the kind of back and forth for this was, to me, there was a benefit in keeping it as simple as possible because I want it to be something that can be easily applied to other things and not become so complicated and esoteric that it won't be applied to other data sets. And capture the most of the pattern that we can with minimal complexity. Just again from a pragmatic kind of useful standpoint. But I think that makes a lot of sense because those different pelagic fish definitely have different efficiencies in terms of growth, and that affects actually. Probably no one knows more about that at this point than the people who farm those fish because they pay attention to that stuff a lot, which helps because you can get a lot of those parameters. Yeah.
I don't think I understood one of the things you said right here at the end about yellowfin tuna, which seems like a very interesting one to look at given its distribution.
This one?
Yeah, you mentioned different growth rates, which is a problem, but growth rate is something that can be estimated from working with analysis.
Right.
And I assume that's been done.
So what I meant was, okay, if before, so we have an equation that takes growth rate into account, right? But a lot of studies didn't have that or don't do that. So they would go and take fish from 50 to 100 centimeters in Hawaii and compare them to fish from 50 to 100 centimeters in the Maldives. And then they'd say they corrected for size, probably based on some exponential relationship. And then they'd say, well, this one's higher than the other one. There's a problem. That one that's higher grows slower in the Maldives than the one in Hawaii. how do you know if that had an effect at all, and how do you know if that's half the effect? How do you know if that's all the effect? You don't unless you can put all the variables together. That's more what I was saying. Ann?
Sort of a off-topic or tangential question, but we know a lot about cognitive and developmental effects, particularly in children, mothers with mercury body burdens. Is anybody looking at these fish?
Pelagic fish? I don't think so.
Any fish.
Yes.
It seems you in a high mercury fish, you should, they should be bumping into walls, having lymph fins, and their larvae should not be able to catch food or something.
Yeah, I have. Yeah, I haven't. I looked into it a little bit, so I don't have a great idea of broadly.
I haven't seen anything, but I haven't really.
No, there have been studies where they have small fish in tanks, and they see how well they predate, and they actually find clumsy predation at high mercury levels. And I think people have looked at. I think that's pretty cool. That's behavioral stuff, and I think people have looked at some other things as well.
My guess is the fishery is driving the population level of these fish, so there would be any discernible contaminant effects going on that would, we could see above that.
Yeah, it's interesting you say that because I've talked to someone, actually the person who's the author of, or co-author of some of the mercury stuff I showed. She's at Harvard and has talked about, these giant scale models that can model fisheries effects and can model everything from atmospheric to deposition to phytoplankton to zooplankton, which are a lot of the pieces that are missing if you want to look at the whole picture, and see if there's an impact on fisheries themselves. And I have to admit, from my background, to imagine that there's an impact on fisheries that even comes close to that of human exploitation is hard for me to swallow. But does that mean it doesn't matter? I don't know. Does that mean it's not interesting? I don't know. But it is an interesting concept.
On that point, there have been studies in freshwater fish, and they suggest that freshwater fish are quite susceptible to mercury at these concentrations, even lower concentrations. But it really hasn't been studied properly in marine fish at all, as far as I know. So we have a student maybe working on that in terms of response of fish brains, basically. If you know how much mercury actually gets into the brains of these fish, and we can, but we don't know. So we know that mercury acts as a nerve poison, but we don't know whether or not these fish will respond to stimuli in the normal way as a function of the concentration of mercury in fish. So it's something to explore.
Yeah, for bluefin, things like homing, their migratory capacity, being able to return to their spawning grounds across the entire Pacific, or predator avoidance, deep diving, stuff like that, be really cool. I mean, the difficulty of working them in captivity, it's impossible to describe. Anyway, yeah.
If I understand your argument correctly, what you're saying is that as a fish gets close to its asymptotic size, in a non-Bartolante sense, it will start accumulating more mercury, correct? And you use K as a correlate, but K is the rate at which a fish approaches its maximum size. It's not, so it's a little bit deceptive. So what I want to ask is, have you tried plotting the data as a function of how far an individual is away from its asymptotic size, rather than using K?
I haven't, and I think that's a good idea. One of the next things I have to do for this is, I don't know if you're familiar with Greg Kaye's work, but I met him when I was in California, and when I went looking up, I have to be honest, I didn't use von Bertalanffy growth equations in my life before trying to do this, right, and get an idea of relative growth. And when I looked it up, I found, the first thing I found was Greg Kaye's tutorial on it. And some of it's counterintuitive. It's like some of the slower growers, like the swordfish, have such a high maximum size that actually their relative growth early on is high. Yeah. And again, you look at those plots, and it's counterintuitive, and at least it was to me. And one of the, I got in touch with him, and he wants to talk. And I'm going to talk with him because I had a feeling as I was doing this that I'm missing the complexity by using just K as simply as I do. So that's the next thing I want to do.
And the related issue is that there are some fish that are harvested so heavily that no fish ever get close to their asymptotic size.
Exactly.
They're not actually in the population for you to measure.
Exactly. So that's what I was saying. You know, the male, we have fish in our data set that, if they are males, are approaching that maximum size. We don't have, probably in any of those, besides the mahi, this one single individual I showed that's extraordinarily large for a mahi, we don't have those maximum sizes for the females. And it'd be interesting to see, obviously, where those data would look like for the really, really big, approaching their maximum size females. They're just so hard to get. I mean, I don't know if, you know, guys go out trying to catch granders, and it's a huge deal, and they certainly don't kill them, you know. And it's just a rare event, and they get bigger than that. So, yeah.
Just one quick question. I know mercury is really the only important element that anybody worries about, but could this apply more broadly to something other than mercury? Or is there any information on anything besides mercury?
So the same person I talked about who has done the mercury stuff is interested in other toxicants. And we've talked about using the same samples as long as we have enough mass to look at similar things and look at the dynamics of all those. You know, right now, frankly, from a practical level, it was the easiest one to start with. Just make direct measurements myself in the lab. Yeah, I mean, all of those things might be interesting. I mean, one of the reasons mercury I think we focus on also is that is the fact that it bioaccumulates so much. So looking across all these sizes makes it the mechanisms a little more, I wouldn't say interesting, but that you can kind of see how different mechanisms would affect it. I would imagine in fish, things like PCBs, environment plays a big role. And that's interesting too, and these animals are migratory, but you might miss a lot of stuff just because you can't control for their migratory history. You can't know their migratory history, so that'd be one complication there. I'm sure that's affecting the mercury as well, to some extent. Carl.
You took us through that interesting parabola with the initial fast growth and growth solution. So why does it disappear in this?
Okay, so that was why it's become clear that's what's nice about the captive data, right, is that it provides parameters, but what's tough about it is just the way it worked out. It's confusing. It's not intuitive of how it works. So this is wild. Now, let me go to where it's big. So this is wild data. This is no longer modeling it. Is this what you're talking about, or are you talking about when we did the extrapolations?
No, you had the nice parabola.
Right.
The growth solution.
Mm-hmm.
So it stops going as fast and then it goes up, but you don't see that reflected in the overall box.
Right. So what?
So does it not have? Am I supposed to be confused?
Yeah.
So I got it right.
No, you're not supposed to be confused in the sense that I'm supposed to explain it better. So basically, the tank, even though their growth rates are mimicking those of the wild, similar to those in the wild, they're not natural conditions, right? So when the fish came in, they were probably feeding. I feel definitely about this now. They were feeding on prey in the wild that were higher than what they were fed in captivity. So their growth, it wasn't that their growth rate increased in the tanks compared to what it had been. It was that their mercury accumulation with growth in their wild conditions before the tanks had already caught up, right? But then when placed into new conditions with lower mercury, growth once again took over. And it took time for that mercury to, the linear mercury accumulation to again counteract that biodilution. So in theory, if they had come in, what we fed them was, for whatever it's worth, 0.12. And if you play with the number, 0.12 parts per million. If you play with the numbers and you feed them 0.3 to 0.4, 3 to 4 times, which is fairly consistent with the Western Pacific, that dip doesn't happen. You get a kind of consistent level, and then it starts to rise. And that's really what the wild data looks like for bluefin. Here, I mean, that's a slight increase, right? But then it really starts to increase. And especially in the Atlantic bluefin, that's what we saw. It's not like the increase is consistent. It's almost flat, and then you see a pretty precipitous rise. So what I think is, the drop is a product of captive conditions, and the part of those captive conditions is that they're eating lower mercury prey than they would in the wild.
And the tilefish that you forgot about, they just grow really slow, is that the answer?
The reason I didn't talk about the tilefish is because applying this stuff, I wanted to keep the highly active open water fish. Tilefish hang out near the bottom, and they're pretty, I don't know if they're constantly moving. I actually don't know that, but
They live with their head in their burrow.
okay, so no.
Down. Right.
Yeah, so there's a lot of stuff going on there. I mean, they're at close contact with the sediment, that means. I don't know much about their diet, and I do think they grow slow, and I do think they're slow growing, and again, I don't know much about their trophic level, but I would imagine that a tilefish that's fairly large has been growing for a long time. So another, I think, fairly similar comparison is like grouper. Grouper are high in mercury too. Why tilefish beat everything else? I don't know. I don't know why they're so much higher, because you didn't see grouper on that chart,
I might have an answer for that. The tilefish data that are out there, especially if it's from Elsie's paper, is based on 2 data points from the 70s.
It's still based on the data?
From what I know, yeah. So it's not, I wouldn't really put a lot of faith in it.
Okay. So I think Josie left, but she was supposed to cut me off a while ago. Sorry, guys. Someone else wants to give me the hook, but thanks for coming.